Thank you, Anne, for that very kind introduction, and thank you for the music up the back. So I want to start this story, sort of Anne gave you a little bit of that history, but I want to go back even further before that to actually when I started in paediatrics as a resident. It's getting longer and longer ago, actually. In fact, you can see how long ago it was because that was the phone I think I had at the time. And you actually used it to make phone calls, which is interesting. And you certainly couldn't Google anything because actually Google, I don't think it had actually been invented yet. It was just, people were just trying to work out what it was. And it was around that time, as I said, I started in paediatrics. My first day on the ward at Sydney Children's Hospital was actually on the cancer ward. And I was pretty worried about this, having come from adult medicine, what it would be like and how depressing and lonely and sad, it would be and whether I would cope with it. But I was surprised to find that it was actually more like this. It was a happy place. There were kids playing, there was music, there was laughter. And actually, it was really the two main reasons for that. One is at the time, and what has continued to be, it was the kids that were really inspiring because even in the face of cancer, they didn't worry and dwell on their future. They just, as long as they felt well, they wanted to get on with it and play. And they were and continue to be the inspiration. It was also not such a sad place because actually most kids with cancer do well. As you heard from Michelle, the cure rates for cancer, have continued to improve. And when you go back, even before that to the 1950s, to illustrate how far we've come, this is actually a young girl who was 3 years old when she was diagnosed with leukaemia. This is actually the daughter of George Bush Sr. and the sister of his son George Bush Jr. And she was diagnosed at the age of 3 with leukemia. And when she was diagnosed in the 50s or 60s, they were told, there's nothing you can do. This is an incurable disease. Take her home. There is no treatment. There is no cure. And in fact, to sort of illustrate what it was like at that time, you look at a quote from a haematologist who said, there's nothing worse than to make a diagnosis of leukemia. The family is deluged with suggestions, and in the meantime, the patient goes steadily downhill. Much as one hates to admit it, there is practically nothing to offer in the acute or rapid case of leukemia. So this is the only graph I'm going to show you, but it's a really important one because it really shows how far we've come since that time with an incurable disease. In the 1960s, kids started going into remission, and gradually, year by year and decade by decade, and part of that is actually the work that was done here at the Children's Cancer Institute, has gradually led to over 90% of children with leukemia, with ALL, the most common type of leukemia, being cured. And it's truly one of the greatest success stories in modern medicine. And it actually started with people in the lab back in Boston where I did training, starting to grow leukemia cells and finding drugs that actually killed them, and then working out how to get these into patients. But as you heard, brain cancer is a different story, and brain cancer is now the single disease responsible for the most deaths in childhood, and so that's what we have been focusing on. And in particular, the worst of all brain tumours, in fact the worst probably of all cancers to affect either children or adults, is one called DIPG, for which there is no treatment, there is no cure. And in fact, if you take that quote from the 1950s or 40s, which sounded very quaint and hard to actually imagine, and you take out the word leukemia and you replace it with DIPG, there is nothing worse than to make a diagnosis of DIPG. The family is deluged with suggestions, and in the meantime, the patient goes steadily downhill. Much as one hates to admit it, there is practically nothing to offer in the acute or rapid case of DIPG. Those words that were sort of quaint and old-fashioned actually still are true today for this disease in 2021. And in fact, another famous person, this is Neil Armstrong, whose daughter had DIPG, and it's remarkable to think of how far we came for one child with leukemia, whereas in the same period of time we really have not made progress with this brain tumour. Even though we got a man on the moon, this man, still we could not cure his daughter today. And one of the problems with DIPG is that it's in such a critical part of the brain that you can't remove it because that would be fatal. And in fact, for decades, people weren't even able to biopsy it. It was felt to be too dangerous. It was in the most critical part of the brain. So we never actually had the tumour to study in the lab. No one anywhere in the world was actually doing any research on it. Certainly no one here in the Institute, no one in the whole of Australia. And so the only way we could actually make a difference when we started this program a few years ago, we thought we need to actually allow parents to have their child's tumour donated after their child's passed away. And we set up a national program to allow this. And our idea was that we would, we had all this fancy equipment that had just been delivered to the Institute, these robotic machines that could actually screen thousands of drugs. And we would use this machine, we'd grow the cells, and we'd use this machine to see, is there anything that's actually active against this deadly disease? So when we came up with this idea, we were told, no, it's impossible. No one's done it before. You probably can't grow these cells, like after a child's passed away. It's not actually going to be possible to grow living cells in the lab. Our grants were rejected. It's just not something that's achievable. And it was actually the parents of a couple of children, Benny and Amy, who said, well, just like we thought, maybe it's not impossible, it's just that it hasn't been done yet. And they actually gave us the first amount of funding and actually helped us get the ethics approval. We had to go to the ethics committee and say, parents want us to do this. It's an ethical thing to do. And we actually this is actually the photo of the first time we were actually able to grow these cells in the lab. The first time actually anyone in Australia had grown these cells in the lab. We went on and actually have developed mouse models that actually mimic in the mice exactly what happens in the kids. So we were able to actually now for the first time start testing drugs and see if we could find any cures. And we actually screened, as I said, as we planned, we ended up screening thousands, over 4,000 different drugs to see if we could find anything that was active against this disease. And actually, almost nothing was. Out of the thousands of drugs, it was only a handful that actually did anything. But we looked at that handful, and one of them actually came from this plant called the feverfew plant, and it was a natural compound called parthenolide. And we actually ended up working with a company who was actually making a synthetic form of parthenolide, a brand new drug called ACT-001, which was actually they were developing as a new anti-cancer drug. And so we started testing in the lab, and actually because they were actually already doing trials in adults, we very rapidly actually went and have now started doing a trial in children and have actually started treating the first children in the world with any form of cancer with this drug, including children with DIPG. And this is actually the slide, the image from one of the patients who is actually on the treatment right now, where you can see his tumour on one side before he started treatment, and it's actually improved dramatically with this drug. And this is a disease where we've never seen that before. This is a disease that doesn't respond to any drug. There's never been any chemotherapy or any other treatment that does anything for this. And for the first time, we're actually seeing a patient, not only with an MRI that's improving, but the patient's improving as well. We have other lead compounds that we've worked on. One of the other things that we've found on from this screening that we've done is that it was actually anti-malarial drugs. Totally unexpectedly, actually 3 different ones were actually very effective against these tumours. And again, many years of research looking at that, and again using a new drug, CBL0137, that's actually been developed from these anti-malarials. We've been working with leading groups around the world and are actually about to open our world first clinical trial of this drug. And actually this is in collaboration with the leading cancer centres in America through what's called the Children's Oncology Group, and this will actually be the first time that a trial like this is open in Australia as well as in America. Not only open in Australia, but actually being led from Australia. So this is exciting, not just for this new treatment, which we hope is extremely effective for setting a new, changing the paradigm where actually Australia is leading in this. Another discovery that we've made and just published is actually we found a completely new way of treating DIPG using this target called polyamines. And what we found is that polyamines are these drivers that DIPG is absolutely reliant on, and without them, it can't grow. And again, we're working with a company that's made a completely new drug that actually blocks the polyamines getting taken up into the cells. And we found that with this combination of treatments where we block the polyamines being synthesized and a second drug where we block them being pumped into the cells, it's actually, in our animal models, it's the most effective treatment that anyone has ever studied anywhere in the world. And as we've just published it in a very high impact paper, and actually it's getting now international attention, again with an international trial to follow after that. And then finally, the other very exciting development is, as Orazio mentioned, using the immune system. And so we're working with Orazio and with others who are experts in this area, where we're actually taking a child's T cell, their own immune cell, engineering it to actually attack the cancer cell, the DIPG cell, and then injecting it back into the child to allow it to then go and attack the cancer. And so this is a trial that again will be opening this year. It'll be the first time in Australia that we've ever had this type of therapy, and for the first time we're hoping that we may actually see a difference for these children. So this is actually just part of the team. This is a little bit old, this photo, but this can only be done with an amazing team of scientists. And everything I've talked about, it's not me who's doing it, but it's all the people who've really dedicated their lives to working in this group to help improve the outcomes for these kids. And with their effort and with what you've seen today, it really for the first time feels like we're starting to make a difference. For the first time, it feels like where there was no hope, we're starting to get some hope. For the first time, we're seeing patients where we'd have no treatments starting to respond to treatments that we've developed here. It feels like we are trying to climb Mount Everest. This is the Mount Everest of all cancers. There is nothing harder or higher. It feels like we are at least at base camp now, and we have the team and the skills and the know-how, and that we will get to the top. Personally, I would never climb Mount Everest. I think it's a pretty stupid thing to do, but this Mount Everest is one that we will climb, and this is the reason why. It's these kids, Benny and Amy, and Finley and Zane and Isabella and Marcus, and Amity and Isaac and Gemma, and Levi and Daenerre, and too many others. And these are the kids that inspire us. It's their parents who tell us we have to keep going. It's them who've raised the money to allow us to get to where we are now. And in the most, biggest act of generosity in some cases, donated their child's tumour even after they've passed away to allow us to do this research. And this is, these are the kids we think of, and this is why we keep going. And I think, people often, as desperately sad as this is, and people ask, how do you do this? And I think the message for me is that as sad as this is, it's also, it's something that I've learned about humanity and the human spirit. And that in the face of this most awful of disease, in the face of the most devastating thing as a parent that you can imagine, we don't cower in front of it. We don't allow this nasty, evil thing to defeat us. But it's the human spirit that allows us to work together to make a difference, to find new hope, and to make a difference for these children and for all the children that we know are there everywhere in the world and in the future. So I really have to thank everyone who's made this possible, the researchers in the lab, the parents, and of course the children. So thank you.